I would like to begin by thanking the organizers for giving me this opportunity to present my work today. I am Maji. I am a PhD student at Department of Physics at IIT Bombay. What you can see in this picture is a contact map, and these red squares in the contact map are the topological associated domain. And these stats are formed by a mechanism which is called loop extrusion mechanism, where this extrusion complex binds to the DNA and then it starts move along the DNA. And whenever it meets a appropriately oriented CTC of protein, it will stop its motion. And in the process, a loop of certain size is formed. Now I will briefly tell about the motion of these SMC proteins along the DNA. So this is an experiment done by Stigler group where a lambda phase DNA of S compound was fed out at protein and then cohesin was incubated. And in this picture, what you can see, this x-axis represents the position of cohesin along the DNA and y-axis is x-axis is time. And what you can see that the motion of the cohesin along the DNA is purely diffusive, it's purely random. Now what they wanted to show is that how does a nucleosome affect the motion of cohesin. So they put a single nucleosome on the path of the cohesin, and so here this arrowhead is the position of the nucleosome along the DNA, and what you can see that the motion of the cohesin in presence of a single nucleosome is hindered, but once in a while it can pass through a single nucleosome. Like here it has passed through a single nucleosome. Here again it has passed through a nucleosome. So so the motion of cohesin in presence of a single nucleosome is hindered, but once in a while it can pass through a single nucleosome. But when a high density of nucleosome was placed, the motion of cohesin is significantly hindered. It almost not moving. It almost static. So they tried to calculate a diffusion rate from this experiment, and they calculated a diffusion rate for high nucleosome spacing. That means for known nucleosome density. And then they did some analysis and estimated the diffusion rate for high nucleosome density. And the estimated diffusion rate was 7 kbps per hour in case of high nucleosome density. So the estimated diffusion rate is 7 kilobits per hour, and duration of metaphase when these large loops are found is around 2 hours. So the estimated loop sizes would be 14 kilobits per. But the loop sizes observed in high CXP when during metaphase are around 250 to 500 kilobits per. So the puzzle is how do these large loops are formed with this low diffusion rate? So we have tried to answer this question. So this is our model. We have modeled the DNA as a one-dimensional lattice, and this cohesin complex has a two random walkers, which are represented by these two rings. And these green electrical fields are the nucleosome, which are placed on the DNA. And these two red flags are the two absorbing boundaries at two ends of the lattice, which are mimicking the role of CDCA protein. So whenever a random walker reaches to at one end of the lattice, it gets absorbed. And the random walker hops to an empty lattice site with a key, and it can hop across the nucleosome barrier with a right Q. And in our simulation, Q is less than P. So at any instant time, the length of the loop is the distance between the two random walkers. And we are interested in the question that how much time is required to form a loop of length L. That means when both of the random walkers are absorbed at the two absorbing boundaries. So plotted the loop formation time, that is the time required to form a loop of length L as a function of Lincoln length. And as you can see here, that as Lincoln length decreases, the loop formation time increases, and that is understandable because as Lincoln length decreases, that means the number of nucleosomes on the lattice increasing. So that means the random walker has to face more number of nucleosomes now. So that's why the loop formation time increases. But what is remarkable is that the loop formation time reaches a maximum when Lincoln length becomes equal to the size of the nucleosome. That is around 150 square. And when Lincoln length is decreased further beyond 150 square, the loop formation time decreases. So the reason of this non-monotonicity is that there is an entropy associated with the hopping of the random walker in the linker region, and this nucleosome acts as energy barrier to the motion of the cohesin. So there is a free energy associated with it, and the free energy barrier is maximum when linker length becomes equal to the size of the nucleosome. So that's why the loop formation time is maximum when linker length becomes equal to the size of the nucleosome. And when linker length is decreased beyond 150 square, that means for smaller linker lengths, the attempt rate to cross a barrier that is increasing. So that's why the loop formation time decreases. These are the three trajectories of the random walker for three different values of Lincoln length, and the non-monotonicity loop formation is also reflected here. The rate trajectory is for delta equal to 150 square, and you can see the motion of the random walker in this rate trajectory is very slow, is almost static. So that's why the loop formation time is maximum in case of delta equal to 150 square. Whereas for other two trajectories, the motion of the random walker is comparatively faster than the 150 square. Okay, so next we have considered the nucleosome wrapping and unwrapping kinetics with DNA. So now the nucleosomes are not tightly fixed to DNA. They can bind and unbind to DNA with some certain rates. So nucleosome, we have kept the nucleosome binding rate fixed at 12 per second, and we are varying the nucleosome unbinding rate. So in a steady state, the nucleosome density is set by the K of. So now if we plot the loop formation line, the loop formation time as a function of Lincoln length in case of dynamic nucleosome, which is represented by this red curve. So you can see this red curve consistently lies below the black one, which is for static nucleosome. So the loop formation process in case of dynamic nucleosome is faster than the loop formation process in static nucleosome. So next, what we wanted to see that this speeding up in loop formation time in case of dynamic nucleosome, whether that is because of the dynamicity of the nucleosome itself or statistical positioning of nucleosome has any effect or not. So it is well known that stochastic binding and unbinding of nucleosomes results in an oscillatory occupancy profile around the fixed boundary protein, which is called the statistical positioning of nucleosome. And here this black oscillation represents the statistical positioning of nucleosome in case of a 10 kilo square finite lattice, which has two fixed boundaries. And in order to remove this statistical positioning, we have made the finite lattice a periodic one. And in case of periodic lattice, there are no boundaries, so that's why statistical positioning is absent. So you can see this red oscillation where there is no statistical positioning, which is for a periodic lattice. And same is true for a 3 kilopascal lattice also. The green oscillation represents the statistical positioning in case of finite lattice, and the blue one is absent of statistical positioning in case of periodic lattice. So now we have plotted the survival probability of the random walkers to survive up to a certain instant time. This black line is the survival probability in case of finite lattice, and the red one is the survival probability in case of periodic lattice. And you can see that the survival probability in case of periodic lattice is higher than the finite lattice. And we have calculated the blue formation time by integrating the survival probability. And you can see that the blue formation time in case of periodic lattice, when there is no statistical positioning, is higher than the blue formation time in case of finite lattice. The conclusion from this part is that though the speeding up in case of dynamic nucleosome largely comes from the dynamicity of the nucleosome, but the statistical positioning of nucleosome has a subtle effect in speeding up the loop formation time. Okay, so at the end, these are the conclusions from my talk. So study the loop formation time of a given loop length by cohesin in presence of a barrier nucleosome. T loop shows a non-monotonicity as a function of Lincoln length, and DNA binding unbinding kinetics enhances the loop formation rate by cohesin. And statistical positioning of nucleosome has subtle effect in speeding up loop formation time. At the end, acknowledgement. So Mithun Kumar is my guide, and Ranjit is our collaborator from the ISST technique. Okay, thank you. I'd like to the question now.
Okay, so thank you very much. Nice talk. So maybe I have a question. So do you consider uh, the linker lengths are a little bit high, no? Because in vivo you have linker lengths of 40 or 50 base pairs, no? So why? And the other question is, can you consider in your model some steric effect coming from the fiber structure?
So
When
we have not considered any steady effect.
okay,
Yeah.
okay, so thank you. So another question, Daniel.
Yes. Hi. I have a question regarding the. So here you were assuming in this model that um, cohesin was diffusing along the
Yeah.
chromatin.
Yeah. Yeah.
So now there is evidence that you have loop extrusion. Really? So how do you think your work will somehow fit in this picture of loop extrusion instead of diffusion?
Yeah. So here in this model, we have not considered activity. This is purely in the picture of diffusion. But we have done some simulation now in case where we have considered the activity, and there also the non-monotonicity in loop formation time exists.
Okay, but
Does that
yeah, but in your model also you do not consider DNA looping in the formation of loops. I think it's just 1D, the 1D diffusion, right?
yeah, 1D diffusion.
So how do you think the fact that you are in loop extrusion, you are forming loops? Do you think it will
So
change the picture?
so basically those two we have considered a loop formation for a fixed length of loop, fixed length of loop. Yeah.